Good morning, students. Our today's topic is principles of treatment. There are two ways to treat an infectious or communicable disease, and these are to reduce the effect of the diseases and to kill the cause of the disease. So let's start with the first one, how to reduce the effect of the disease. Students, it can be done by providing symptomatic treatment. We can provide treatment that will reduce the symptoms, which are usually because of inflammation. For instance, we can take medicine that bring down fever, reduce pain or lose motion, etc. We can also have bed rest to conserve our energy. These steps will enable us to focus on healing. However, such a symptom directed treatment by itself is not sufficient, as it will not remove the disease causing microbe from the body. So, to cure the disease, we need to kill the microbes. So, now how to kill the cause of the disease? For example, pathogens. Student, the most common method to kill disease causing microbes is to use medicine that kill microbes. Now we have to choose a specific drug that is effective against a particular group of microbes. I hope you remember that we have earlier studied that disease causing microbes are classified into different groups such as viruses, bacteria, fungi and protozoans. Each of these groups of microbes have some essential biochemical life processes. And these processes may be for the synthesis of new substance or for the respiration. But our cells have different pathways than the ones used by these microbes groups. So therefore, we find a drug that blocks, right? So for example, the bacterial like anabolic pathway without affecting your own. And this is what is achieved by antibiotics. Similarly, there are drugs that kill protozoan, etc. So definitely we should know about the antibiotics. Student, antibiotics are actually chemical secreted by microorganisms that kill or hinder the growth of certain other kinds of microorganisms. Sir Alexander Fleming in 1928 first discovered penicillin. He noticed that an agar plate inoculated with the Staphylococcus bacterium had become contaminated with a mold. He found a clear zone in the agar plate devoid of bacterial cells. And the mold was named penicillin. Fleming called his inhibitory substance penicillin. And after this discovery, the development of antibiotics began. Now, some other known antibiotics include streptomycin, tetracycline, chloromycetin, etc. Now, our next question is why it is difficult to make an antiviral medicines than antibacterial drugs. This question is really very important nowadays, as we all are suffering from a viral disease corona and still we are waiting for a medicine for this disease. Student, it is really difficult to make antiviral medicine than antibacterial drugs. Now, one reason is that viruses have few biochemical mechanisms of their own. They enter host cell and use host cell machinery for their life processes. And this implies that there are relatively few virus-specific targets to aim at. But despite these limitations, effective antiviral drugs are available. For example, drugs to keep HIV infection under control.